Think about a conventional weapon. It's loud, it's obvious. You hear the explosion, you see the destruction. There's no mistaking when it's been used. But what if there was a weapon that announced nothing? A weapon that's not being built in a secret military base, but in university labs and biotech startups all over the world. This weapon is silent. It's invisible. It could spread with just a simple handshake or a cough in a crowded room. It waits, it multiplies. And by the time anyone even realizes what's happening, by the time the first symptoms show, it's already everywhere. It has been for weeks. This isn't science fiction. It's the potential reality of bioweapons in our modern, interconnected world. What do you think is the best way to prepare for such a threat? Let me know in the comments below. And don't forget to subscribe for more deep dives into the future of technology and security. Thanks for watching. There is a category of threat that the human immune system was not designed to recognize. Not because the threat is new in any biological sense. Pathogens have existed for billions of years. They have shaped the evolution of every complex organism on Earth. The arms race between host immune systems and the microorganisms that exploit them is the oldest and most consequential conflict in the history of life. Our immune systems are, in many respects, extraordinarily sophisticated, the product of hundreds of millions of years of evolutionary pressure from exactly the kinds of threats they are designed to detect and destroy. But evolutionary pressure works on timescales of generations. It produces defenses against threats that have existed long enough to select for resistance. It does not produce defenses against threats that did not exist until last year. Against organisms that were designed in a laboratory by an intelligence that understands the immune system's detection mechanisms and has deliberately engineered around them. This is the specific and novel danger of synthetic biology. Not that it creates pathogens. Nature has always created pathogens. The danger is that it creates pathogens faster than evolution can respond, faster than immune systems can adapt, faster than public health infrastructure can detect, characterize, and counter. And potentially, this is the part that the biosecurity community discusses with a particular kind of careful quietness, faster than civilization can survive. To understand where we are, you have to understand how we got here. The history of molecular biology in the 20th century is a history of accelerating capability. The discovery of the double helix structure of DNA in 1953 was the beginning. The development of recombinant DNA technology in the 1970s, the ability to cut DNA at specific locations and insert foreign genetic sequences, was the first major inflection point. For the first time, organisms could be modified in directed, intentional ways. Genes from one species could be introduced into another. The genetic material of living organisms was no longer read-only. It had become, in principle, editable. The practical applications of this capability were initially limited by the difficulty of the work. Sequencing DNA reading the genetic code of an organism required large teams, expensive equipment, and years of effort. Synthesizing DNA writing new genetic sequences from scratch was even harder. The tools existed in theory long before they existed in practice at any useful scale. Then the tools improved dramatically, continuously, on a curve that the biotechnology industry tracks with the same kind of attention that the fantastically computing industry once gave to Moore's Law. The first human genome was sequenced between 1990 and 2003. It required an international consortium of laboratories, an investment of approximately $3 billion and 13 years of work. By 2010, the cost of sequencing a human genome had fallen to approximately $50,000. By 2015, to $1,000. By 2022, commercial services were offering whole genome sequencing for less than $200, with results returned in days. The cost of DNA synthesis, of writing genetic sequences rather than reading them, has followed a similar trajectory, declining by orders of magnitude over the past 2 decades. What once required specialized equipment available only in well-funded research institutions can now be performed with benchtop instruments that are commercially available, relatively affordable, and increasingly automated. And in 2012, a technology arrived that changed the landscape completely. CRISPR-Cas9 is a molecular tool derived from a bacterial immune system. Bacteria, like more complex organisms, face the threat of viral infection. Over billions of years of evolutionary pressure, some bacteria developed a mechanism for recognizing and destroying viral DNA, a system that stores fragments of previously encountered viral sequences and uses them as guides to locate and cut matching sequences in new infections. Scientists Jennifer Doudna and Emmanuel Charpentier, working independently and then collaboratively, recognized that this bacterial immune mechanism could be repurposed as a general-purpose genetic editing tool. The CRISPR system can be programmed with a guide sequence, a short RNA molecule that matches a target DNA sequence, and will locate and cut that sequence with high precision in any genome. Deliver this system into a cell, and you can edit the genome of that cell at a specific location. Delete a gene, insert a new sequence, modify a single base pair out of billions. The implications were immediately recognized by the scientific community. Doudna and Charpentier received the Nobel Prize in Chemistry in 2020, less than a decade after publishing their foundational work, an unusually rapid recognition that reflected the magnitude of what they had discovered. But the implications that the scientific community recognized were not only the beneficial ones. CRISPR made genetic editing accessible, not just to well-funded research institutions with specialized expertise, but to any molecular biology laboratory with standard equipment and a graduate student who has completed a semester of coursework. The protocols for basic CRISPR editing are published, freely available, and taught in undergraduate biology courses. The reagents are commercially available. The equipment required is present in thousands of laboratories around the world. The democratization of genetic editing technology is real, and it is accelerating. And democratization, in the context of a technology capable of modifying the properties of pathogens, means something specific and consequential. It means the number of actors capable of engineering a dangerous pathogen is no longer measured in nation-states. It is measured in individuals. Let us be precise about what engineered pathogen enhancement actually means, because the concept is often discussed in terms that are either too abstract or too sensationalized to be useful. A pathogen's danger to human populations is a function of several properties. Its transmissibility, how easily it spreads from one host to another. Its virulence, how severe the disease it causes in infected individuals. Its incubation period, the time between infection and the appearance of symptoms, during which an infected individual may be spreading the pathogen without knowing they are infected. Its immune evasion, how effectively it avoids detection and destruction by the immune systems of its hosts. And its host specificity, the range of species it can infect. Natural evolution optimizes these properties through random mutation and selection. A pathogen that is more transmissible spreads more successfully. A pathogen that evades the immune system more effectively survives longer in its host. But natural evolution is blind. It optimizes for the pathogen's survival and reproduction, not for any particular combination of properties. And some of the most dangerous combinations, high transmissibility combined with high virulence, for example, are actually disfavored by natural selection because they kill their hosts too quickly to spread widely. Directed engineering does not have this constraint. A molecular biologist who understands the relationship between a pathogen's genetic sequence and its phenotypic properties, its transmissibility, its virulence, its immune evasion, can, in principle, modify those properties in directed ways. Not randomly, not through the slow accumulation of mutations over many generations, but deliberately, by identifying the genetic sequences responsible for specific properties and editing them to produce a desired outcome. This is not theoretical. It has been demonstrated. In 2011, two research groups, one led by Ron Faucier in the Netherlands, one by Yoshihiro Kawaoka in the United States, independently engineered variants of H5N1 avian influenza that were transmissible between ferrets through respiratory droplets. H5N1 in its natural form has a mortality rate of approximately 60% in confirmed human cases, but spreads poorly between humans. The engineered variants combined the high mortality of natural H5N1 with the COG1 with the high respiratory transmissibility of pandemic influenza strains. The research was conducted for legitimate scientific purposes to understand the pandemic potential of avian influenza and to prepare countermeasures. The results were so alarming that a major scientific journal initially declined to publish them in full, pending a security review. The point is not that this research was irresponsible. The point is that it was done. That the technical barrier to engineering a highly transmissible, highly lethal respiratory pathogen was crossed in an academic research setting, using techniques and equipment that are now considerably more accessible than they were in 2011. Now consider what has happened to those techniques and that equipment in the years since. CRISPR has become faster, cheaper, and more precise. DNA synthesis has become more accessible. The bioinformatics tools that allow researchers to predict the functional consequences of specific genetic modifications have improved dramatically, driven by advances in machine learning applied to protein structure prediction. AlphaFold DeepMind's protein structure prediction system can now predict the three-dimensional structure of a protein from its amino acid sequence with accuracy that matches experimental methods, a capability that has profound implications for understanding how specific genetic modifications will affect pathogen behavior. The combination of accessible gene editing tools, affordable DNA synthesis, and powerful computational prediction tools means that the technical barrier to engineering a dangerous pathogen has fallen significantly. It has not fallen to zero. The gap between theoretical capability and practical execution remains significant, and most individuals with access to the tools lack the specific expertise required to use them for this purpose. But the gap is narrowing, and the direction of travel toward greater accessibility, lower cost, and more powerful computational assistance is not reversing. The biosecurity community has a name for the concern this trajectory produces. They call it the democratization of destruction. The same process that made computing power available to individuals, that made publishing available to anyone with a laptop, that made complex financial instruments accessible to retail investors, is now making the technical capability to engineer dangerous pathogens available to an expanding population of actors whose intentions cannot be assumed to be benign. Here is where the scenario departs from the history of natural pandemics in a way that matters enormously. Natural pandemics are detectable. They emerge from a specific geographic location. They spread along identifiable routes, trade networks, travel patterns, population density gradients. They produce cases that cluster in ways that allow epidemiologists to trace the outbreak back to its origin and model its spread forward. The detection and response infrastructure that the global public health community has developed over the past century, the surveillance networks, the genomic sequencing capacity, the international reporting mechanisms, the emergency response protocols, was built around this model of pandemic emergence. A deliberately engineered and deliberately released pathogen does not necessarily follow this model. An actor with sufficient knowledge and intent could engineer a pathogen optimized for properties that specifically defeat the detection and response infrastructure. A long incubation period, measured in weeks rather than days, during which infected individuals spread the pathogen without symptoms, allowing it to achieve global distribution before the first cases are recognized. Immune evasion properties that cause the initial disease presentation to resemble common illnesses, delaying recognition of a novel pathogen. Genetic sequences that do not match any known pathogen in surveillance databases, slowing identification. Properties that allow the pathogen to survive on surfaces or in environmental reservoirs longer than natural pathogens typically do. And an actor with sufficient operational sophistication could release such a pathogen not from a single location that could be traced, but from multiple locations simultaneously, seeding the outbreak in multiple cities on multiple continents. At the same time deliberately destroying the epidemiological signal that would allow the source to be identified and the spread to be modeled. In this scenario, the first indication that something is wrong would not be a cluster of unusual cases in a remote province that triggers an international alert. It would be a diffuse, globally distributed rise in cases of what appears to be a severe respiratory illness occurring simultaneously in cities on multiple continents, with no identifiable common source and with a genetic profile that does not match anything in the existing pathogen surveillance database. By the time the nature of the threat is understood, by the time the genomic sequencing is done, the epidemiological picture is assembled. The novel pathogen is characterized, the response infrastructure is mobilized, the pathogen would already be everywhere. This is the silent kill. Not silent in the sense that no one becomes ill. Silent in the sense that the weapon has already achieved its effect before anyone knows it has been used. The public health response infrastructure that exists today is, in many respects, genuinely impressive. The genomic surveillance networks built in the wake of COVID-19 are more extensive and more rapid than anything that existed before 2020. The international agreements on pathogen sharing and outbreak reporting, while imperfect, represent a real framework for coordinated response. The mRNA vaccine technology demonstrated during COVID-19 showed that novel vaccines can be designed and manufactured on timescales that were previously considered impossible, weeks from sequence to clinical candidate rather than years. But this infrastructure was built to respond to natural pandemics, and its adequacy against an engineered pathogen specifically designed to defeat it is not guaranteed. The mRNA platform can produce a vaccine candidate rapidly, but only after the pathogen has been characterized. Characterization takes time. And in the scenario described above, the time available before the pathogen achieves effective global distribution may be shorter than the time required. There is also a dimension of the engineered pathogen threat that has no analog in the natural pandemic response framework. Natural pandemics do not have an actor behind them. They do not adapt their strategy in response to countermeasures. They do not have the ability to modify the pathogen in real time as the response infrastructure deploys. An engineered pathogen in the hands of a sophisticated actor with ongoing laboratory capability could potentially be modified in response to emerging immunity in the population, a directed evolutionary process operating on a timescale of weeks rather than the years that natural evolutionary pressure requires. This is the scenario that biosecurity researchers who study this problem most carefully describe as the one that keeps them awake. Not a single engineered release, catastrophic as that would be, but an ongoing, adaptive campaign, a pathogen that evolves not through natural selection, but through directed engineering, staying ahead of immune responses and countermeasures in a way that natural pathogens cannot. The immune system was not designed to defend against an adversary that understands its architecture. The governance response to this threat has been real but inadequate relative to the trajectory of the technology. The Biological Weapons Convention, which prohibits the development, production, and stockpiling of biological weapons, has been in force since 1975. It has no verification mechanism. It relies entirely on self-reporting by signatory states and has never been enforced against a state actor. It does not address non-state actors at all. And it was written before CRISPR, before affordable DNA synthesis, before the computational tools that now make pathogen engineering accessible to a far wider range of actors than the nation-states the convention was designed to constrain. Export controls on dual-use biological equipment and reagents exist in most developed countries, but are porous, inconsistently enforced, and increasingly inadequate as the equipment becomes more widely manufactured and the reagents more commercially available. The biosecurity community has advocated for a range of additional measures. DNA synthesis screening to detect orders for sequences associated with dangerous pathogens, enhanced surveillance for novel pathogen emergence, international frameworks for the governance of dual-use research. Progress has been made. It has not been made at the speed the technology is advancing. The fundamental problem is familiar. The governance response to an emerging threat is calibrated to the threat as it was, not the threat as it is. The Biological Weapons Convention was adequate for a world in which biological weapons required nation-state resources to develop. The world in which individual actors with graduate-level biology training can access the tools required to engineer dangerous pathogens is a different world. The governance frameworks have not caught up. They may not catch up in time. There is a specific and sobering comparison that biosecurity researchers draw between biological weapons and nuclear weapons. Nuclear weapons are physically enormous. They require rare materials, highly enriched uranium, plutonium, that are difficult to produce and heavily monitored. They require sophisticated engineering and precision manufacturing. The barriers to nuclear weapons development are high enough that, in the 7 decades since the first nuclear test, the number of states that have developed them has remained in single digits, and no non-state actor has ever successfully produced one. Biological weapons have no equivalent physical barrier. The materials are not rare. The equipment is not specialized beyond what exists in thousands of research laboratories around the world. The knowledge base is published, distributed, and expanding. The trend in biological weapons capability is the opposite of the trend in nuclear weapons capability. It is moving toward accessibility rather than away from it. This asymmetry is what biosecurity researchers mean when they describe the engineered pathogen threat as categorically different from the nuclear threat. Not necessarily more immediately destructive than nuclear weapons in a single event, but potentially more accessible, more difficult to attribute, more difficult to deter, and capable of effects that propagate through the global population in ways that no other weapon can match. A nuclear weapon destroys a city. An engineered pandemic destroys the population of the planet. Not certainly, not inevitably, but potentially. And the barrier between theoretical potential and practical reality is lower and declining faster than most people outside the biosecurity community understand. The Fermi paradox, examined through this lens, offers a possibility that is difficult to dismiss. The development of biology as a science is not unique to Earth. Any civilization that discovers the molecular basis of life, that develops the tools to read and edit genetic sequences, that produces the computational power to model protein folding and predict the functional consequences of genetic modifications, any such civilization will arrive at some point in its development at the same threshold we are approaching. The point at which the technical barrier to engineering a dangerous pathogen falls below the level that governance and detection systems can reliably intercept. This threshold may be one of the most consistent features of technological civilizations, not because it is inevitable that someone will cross it with malicious intent, but because the number of actors with the capability to cross it grows as the technology becomes more accessible, and the probability that at least one of them will do so, whether through malice, through accident, through the specific kind of motivated reasoning that allows a person to convince themselves that their cause justifies the means, increases as the population of capable actors grows. The universe may be silent, not because civilizations do not exist, but because the transition from biological ignorance to biological mastery is one that most civilizations do not navigate successfully. Because the window between developing the tools to read life and developing the governance to prevent those tools from being used to end it is narrow. Because the silent kill, by its nature, does not announce its arrival. And because the most dangerous morning in the history of a civilization does not begin with a sound. It begins with a handshake. Somewhere right now in a laboratory that does not appear on any watch list, with equipment that was purchased through entirely legitimate channels, a sequence is being designed. Not with malice, perhaps with curiosity, perhaps with the specific kind of certainty that comes from believing that what you are doing serves a purpose large enough to justify the risk. The sequence will be synthesized, it will be tested, it will be refined, and at some point, at some specific, ordinary, unremarkable moment, it will leave the laboratory. It will spread through a handshake. It will wait. And the world will not know what is happening until the world is already different. The question that the Fermi paradox leaves hanging in the silence of the universe is not whether this has happened before. It is whether, on the mornings when it happened, the civilizations that faced it had built anything capable of stopping it in time. The evidence from where we stand is inconclusive. And inconclusive in this context is the most frightening answer of all.